Orthoptera now is focusing on African fauna, and he's the mentor of the Italian orthopterists. Please.
Thank you very much, Filippo, also for inviting me in this section. Now we begin with Paolo Fontana, who will speak on records on the possible effect of climate change on the distribution of some orthopteroid insects. Please, Paolo.
Thank you. So, 15 minutes. I will try to do better. Okay. Thank you very much. I excuse, but probably the best title of this presentation would be What a Feeling, but I'm not Irene Cara. This is not flash dance, but what I will speak about is relative to some feelings, sensation that I obtained by last year's field trips. Since 2009, I work as an apidologist, but I cannot forget the orthopteroid insects. So time to time, I do some field expedition and I produce some small research. So in recent years, I concentrated my research in the northwestern part of Italy, in order to study the genus of the Matera chelidura and Podisma too for Orthoptera, and in central Apennine for Italopodisma and Forficola pseudo-chelidura. About the genus Podisma, the species Podisma pedestris is very interesting for me because there are some populations that in the past have been described as separate taxa, especially Podisma pedestris caprai from Alpe Fenestre in a very nice place, and Podisma pedestris nadigi from Passo Croce Domini in Lombardy. And I made some field trips to search these two species, but for Podisma pedestris nadigi, I found a very bad weather, so it was not possible to say how present or not it was. But for Podisma pedestris caprai, I investigated also the literature of the past 10th, and I visited many localities. I arrived to the upper part of the type locality and in a perfect weather condition and so on. And I didn't find any Podisma. And so it was very strange for me because the last paper that worked on this area 40 years ago, there was a lot of specimens in many localities of Podisma. So this was very strange. Also, I made about the genus Podisma some research to search the Apennine Podisma, especially Podisma ruffoi, Podisma goidanici, Podisma, some other Podisma of the Apennine, as you can see here. And some of them are still present thanks to the research of the University of Roma III, but others are really very scarce, while in the literature was collected by many authors. During the expedition in the west northern part of Italy, in the last 4 years, I feel that even the very common Epipodisma pedemontana, I remember one of the first meeting of the Grio group we spent in Piedmont, and we collected a lot of these species in many localities. I found very scarce. At the same time, I observed very small population of Boemanella frigida. As regarding the genus Italopodisma, I visited in the last 5 years quite every year some localities like Gran Sasso, Campo Imperatore, and in comparison with my first expedition that I made in the end of the 90s together with the dear friend and colleague Roy Kleckers, the abundance of Italopodisma is significantly reduced, significantly for my eyes. And what I observed in this case is that a bird, Fringuello alpino, no, I don't remember, Montifringilla nivalis, that was grazing on this Orthoptera in big numbers in these localities, are really reduced in number. So in the same period of the year. For the genus Italohippus that doesn't produce population so big like the Podismine, but are more scattered, more rare, also in this case I observed a strongly reduction. In Gran Sasso, I also searched actively in the last 5 years a very peculiar earwig from Italian fauna. I think it is the most beautiful forficula we have, Forficula appennina, that was known from the highest mountain of central and southern Apennine. I found for the first time this species in Gran Sasso together with Roy Kleckers. Then I found in the end of the 90s with Filippo Maria, and I searched in last 5 years. I didn't found near the localities I knew, Rifugio Duca degli Abruzzi, but I found in a higher, in a more isolated locality on Monte Aquila, where I never found. So are very small data. But what impressed me more about this apparently reduction of high mountain species was the research on the genus Chelidura. In 2019, I made a field expedition collecting all the known localities for this genus in Western Alps, especially from the Italian side, but also some locality in the border with France. And some of these localities was not very precise because there was the name of the nearest village, but other localities was very precise with the altitude and so on. We visited all these places with the yellow sign, but we found Chelidura only in the 2 green circled places. I repeated some of these localities in 2021, and I found again the Chelidura montana in Gran San Bernardo, exactly in the place where have been described the type material, between the hostel of San Bernardo and Lid, and exactly in the same place, a square of probably 50 meters for 50 meters, I found it again. I visited 3 years the locality of Chiappera di Aceglio, from where I have a very long series of specimens collected by Professor Beppe Osella. And it is a new species that we are going to describe in his honor. And we walked all around the valley to all the altitude and so on, and we did not found any specimen. In all these localities where we did not found Chelidura, we found Anecura bipunctata. And only in the two places where we found Chelidura, Anecura bipunctata was not present. So can be also a problem of competition, I don't know. Can be that this species is more adapted to live in different conditions. So what I feel is that the distribution of and the relative abundance of some species from high mountains is a little changed in last year. We cannot say that there is a decrease because we know many species of orthopteroid insects, especially Orthoptera, that has very strong fluctuation in population. We have a very nice example in Italy, for example, Uromenus rijoi, described with 100 specimens in the type material in the 70s, and after that the species have never been found in Sicily. And in Sicily there is some good orthopterologist, if I remember. But the opposite was for Barbitistes vicetinus, that Galvagni and I described on one single specimen. No specimen have been found in any collection collected before of this time, and only after more than 10 times the species became very common, producing outbreaks and so on. So some species are really able to fluctuate strongly in their population, but when we consider species or genera that has a wide distribution, and we can observe the reduction of the population, means that probably something is happening. And in some cases is quite interesting, because for the genus Chelidura that was known to have only one species, Chelidura pyrenaica and Chelidura aptera, now we can say that there are more than one, that there are two species, and on the basis of molecular as well morphological analysis, now we are going to describe two new species, extending the range of the genus. And so single population can be very important by the conservation point of view, because they are probably unique population of some species. So I close my feeling communication suggesting to that as many of you, especially young people, are going to study the old locality from where we have a good list of species, and to investigate again in nowadays to see if there is such kind of problem. And probably as some other species cited this morning by the very interesting presentation, could be chosen as a target species to do some kind of investigation. In high mountain, we have also problem of human impact, tourism, we have seen for Austria and so on, and also for grazing. The managing of the grazing in the mountain is very changed in last half century. But I think that to speak of climate change is probably inappropriate. But when we start to speak of 50 years, probably something concrete can be found if we increase our investigation. Thank you very much.
Thank you. There is time for one question. Question? No?
Tutto chiaro.
Thank you. Now, Ettore Rivalta selected localities for monitoring Orthoptera in Trento province across time. Please, Ettore.
Thank you. Now, I'm Ettore Rivalta, and I will talk to you about the work I have done with Filippo Buzzetti and Jonathan Stanker, and also Costanza Geppert and Lorenzo Marini of University of Padova for this museum. Let's start describing quickly the study area for those who don't know don't know Trentino. Trentino is characterized by various mountain ranges and various that have various features, natural and also geological features, and are divided by some deep valleys, and that we can divide it in two parts. One, as regards Orthoptera fauna, one part is characterized by a fauna, an alpine fauna, and the southern part is characterized by a pre-alpine fauna that is very interesting and various, and there are a lot of endemic and interesting and localized species, and for this it is a very interesting study area, and you see that it's also very well studied by particularly by Antonio Galvagni, who lived in Rovereto and collected many specimens that are collected, stored in this museum. And it is very interesting also the work by the botanist of the Civic Museum, of this museum, that collected in various years many data, and it is a very huge work. And this work describes the situation over time in a diachronical perspective, and you can see how the distribution of some mountain species is changing. For example, many species are going up in higher and higher altitudes. And this is, of course, very interesting for our research. So we focused on 33 localities where in these localities were done samplings between 2008 and 2009, and we go there in last 2 years to sample with the same methods, and considering also, for example, how much time passed between the last mowing and our sample, or if there are trees and bushes, and the conditions of the various localities. And then we send our data to Costanza Geppert and Lorenzo Marini of University of Padova that process this data, and we have some preliminary results that show some trends for these localities. For example, we see an increase of generalist species, like Corcypus brunneus and Corcypus dorsatus and Pseudocorcypus parallelus that are actually everywhere in Trentino, and a reduction of the mountain species, the typical mountain species that 15 years ago were the most common in these localities, like Decticus verrucivorus, Stauroderus scalaris, and Chisella irena. This piece, there is a reduction of this piece. And we also observed the appearance of the Decticus albifrons, that is a southern species which is expanding its areal, and it is very invasive, and now in Trentino is it's common and more common and more common, and yeah. But there is this work is only started because localities are not enough to say to have to make a description that is enough good for us, but. And also, the real question is if this there is a competition between species, or if there is what of these causes is more important, like the competition of the generalist species and the mountain species, or the climate change. And the habitat change, and we will continue to monitor some species that are very interesting of this region, like Pseudoprumna baldensis and Cortopodisma cobelli, that are very interesting endemism of this area. Or, for example, Pseudopodisma fiberi, that in Italy is localized. There are various populations in Friuli, in the east, but we discovered a population in Montefinocchio, here near Rovereto, and this is now the westernmost population of Pseudopodisma fiberi, and we think it is more common. And then, yeah, there is a lot of work to do. Thank you.
Some questions? Axel.
This is an interesting question. We, in our work at Rovereto, we didn't work on this, but there are some works that and seems that these species follow some routes, some anthropic routes, and it is very common. For example, in cities. For example, in my city, Bologna, it's incredible. In the last 3-4 years. in the center of the city, there are incredible population of these giant grasshoppers that terrify the population of Bologna and made this sound that is very strong and metallic, and it's very strange for people. For normal people, but it's a good question, and Dactylus albifron is a very interesting species that, and yes, so we will try to work on this.
Maybe both climate change and expansion induced by man. Yes.
Thank you for a good answer. I don't speak very good English.
Oh, yes,
sorry, I'm not in.
But we understand. Yes. Okay. Ettore is the youngest orthopterist in Italy. He described also one or two species?
No,
one.
No, the last one,
Now we have Filippo Maria Buzzetti, Orthoptera conservation in Italy, Zeuneriana marmorata e ad Nonconotus italo-austriacus. Please, Filippo.
Thank you, Bruno. It's some years that I am focusing on conservation of Orthoptera, and especially on some species that huge, in my opinion. The first was Zeuneriana marmorata. It was thought to be extinct, but in the 90s, Paolo and Roy found one or two population, I think one population. And then started some further researchers that found some more populations. All these populations are small, not in the number of individuals, because some are very dense, but are very localized. And what is most worrying is that they are not connected, they are separated, so there is no interpopulation genetic flow. And so we started with the University of Trier and the University of Padua and the museum. We started analyzing these populations. We chose one suitable habitat without the species. It was known for years, for decades, to be without the species, and we translocated some specimens. And this is the locality. It is we translocated from Isola della Cona to Schiavetti, and this is the locality where we introduced some specimens, some individuals. We started 5 years ago, and it is we choose this place also because it is a place very near to the town, but it has some natural or semi-natural components. This is a fragment of nature in the town, we could say. As you can see in this slide, you have the exact plot where we introduced the first individuals. In the 2022, we found the first individuals after 1 year, and the area occupied by individuals was larger year by year. As you see, we were able to check the local distribution of the species, and we saw until this year an increase of the area of presence. We paired the activity between field activity, where we were focusing on searching individual, recording and trying to confirm the presence in the locality of in the new locality, and we tried to breed some individuals. Breeding, it has been quite difficult because it was not easy to follow day by day the captive individuals. But we were able to have new individuals, some nymphs born in captivity. And so we can confirm that we have we have established a new population of these endangered species in a locality where this species was previously absent. So we would like to repeat this method, this protocol, to another endangered species that is now is a very different, completely different, ecologically different species, because it is Anonconotus italo-austriacus. It lives on high altitude and lives in the high altitude alpine pastures and bushes. We are studying now this population in the Dolomiti National Park that is very isolated. It is in a place that is difficult to reach because are 11 kilometers by walking. You have to walk for 11 kilometers just to make 1,000 meters of altitude gain. And you reach this beautiful place in the Dolomiti that are the remnants of an high altitude glacier. What you see here are the traces, or the holes made by 3 glaciers of high altitude during the last ice age. And they have this strange name, Busa di Cavarin, Busa delle Vette, Busa di Pietena. Well, for those who are not Italian, we will call one 2, and 3. And this is the area where this population lives. I say this population lives here because during the years I surveyed these 3 plots every year, but I found only scattered individuals, 1 or 2. It appears that it is present every year here only, with a high number, sometimes a high number of individuals. Here on the bottom, on the right, you have a population that we are checking if it is, as it appears, extinct, from Monte Baldo or Monte Altissimo that are very near here to the museum. And of these species, we have other 2 or 3 populations in in Italy, but all these populations are geographically isolated. Genetic studies seems to say that this Dolomiti population, this one, is very different from the other. So, what we would like to do, we want to check the distribution, the actual distribution of Anonconotus italo-austriacus in Busa delle Vette, the plot 1, and in the surrounding areas, because we would like to see if, where it is always present. We would like to identify the preferred habitat and the suitable areas for future translocations, because this small population could be a donor population for withdrawal some individuals to introduce in other suitable habitats. We will try to identify the host plant and evaluate the sex ratio, and we will keep the specimens for 24 hours isolated in tubes, and we collect the feculae, the feces of this individual, to check what they eat, because actually we don't really understand. In captivity they eat everything, almost everything, but in nature I never saw some individual eating. And we would like to identify areas suitable for translocations, recording the sound. I will speak of this later. And shoot videos in nature of isolated males, isolated females, and the behavior, the pre-mating, mating, and the post-mating behavior. Well, this is the plot 1, Busa delle Vette, the place where the donor population lives. This is this small hill where the population is every year present, with a fluctuating number of individuals, some years more, some years less, and this is it. It lives in this bush area, not in the grass. It appears that it is only an idea, but in this area they have grazing, they have cows that graze during the summer, and at the end of the summer, but even when the phenology of these species start, you have adult individuals only in this small hill. We don't know if it could be an effect of the grazing, so that, some kind of disturbance that make the specimens, the population, easy to live only in the places where the cow don't go. Here is where they live. They live in the bush, not in the grass, maybe because the bushes are where the cows don't go, don't step. They are very hard to find, but I will show you. One is here and the other is here. They are very difficult to spot, but a trained researcher can find them. Plot 2, Busa di Pietena. It has a suitable habitat, but the species appears to be absent. If this is true, that is absent all over the year and every year, this could not be confirmed by the finding of the last year of 2 individuals here on the edge between plot 1 and plot 2. As you can see, the ridge dividing the 2 plots are very steep, and it could be a geographical barrier that stop the erratic, the walking individuals. So we found 2 individuals in the grass, in a completely different environment, not in the bush. But last year, we found this, last year there was no grazing because there were no cows. Then we surveyed the plot 3, Busa di Cavarin, where there is also suitable habitat, no presence. The only individual was found by me years ago on the top of this mountain. Well, I would not go again on top just to check if there is another individual. I would like to find it here because it's more easy. This is what we are planning to do. We are already doing this year surveys with the University of Padua and the University of Parma. We are also in co-work with Philipp Kirchner and Petra Kranebitter, that we are glad to have here. We are comparing the sound of each species because they did a very huge survey years ago. They traveled all the distributional area of the genus Anonconotus, and they collected each species. They gave me the living specimens and I recorded them. And these are the first result. You can see a general pattern or acoustic pattern, but you can you can see some differences between the species. Strange that you can see that similar songs are emitted, but are emitted by near populations or near species, and there are some species that emit different kind of sound. However, now we are checking the presence on Montebaldo for Anonconotus because since this year the Museum of Rovereto is in head of the managing of the natural park of Montebaldo. So we are planning to check the distribution variation, the high altitudinal variation, if is it any, of Pseudoprumna baldensis that is endemic of Montebaldo, and it has two populations in the Alps of Lombardy. And we would like to confirm the presence or absence of Anonconotus italo-austriacus here. This is the Montebaldo ridge. This will be probably this and the next, the last year that we will survey for Anonconotus italo-austriacus here, because already Galvagni assumed that if there was a population, it could be has extinct because of the First World War that in this area was very intense. So we have also, we are focusing now not more on the sea, on the coast, but on the mountains, because recent studies show that the Alps are a core of biodiversity biodiversity treasure that host many endemic and many endangered species. This probably because the mountains support a high number of endemic species because mountains are less transformed by humans. The anthropic effect is lower than on coasts it could be. So we are now changing from Zeuneriana. I promise I will not talk anymore about Zeuneriana. I will talk only about Anonconotus italo-austriacus. Thank you. Thank you.
Thank you, Filippo. Some questions? Please.
Filippo, I saw, because you said a question about the host plant of Anonconotus italo-austriacus, I saw it feeding on Musa delle Vedette on blueberries.
Yes, yes, but it doesn't eat blueberries. This is strange. I think it goes here because it is disturbed by cows grazing. It is my hypothesis.
No, I saw a female eating.
You saw a film. Oh, thank you. I will steal this data. No, thank you. Thank you very much. So do they... Okay. Blueberry. Blue. And is this one blueberry? Okay. Vaccinium, yes. Okay, thank you very much. Thank you.
Filippo, how many specimens of Zeuneriana marmorata have you left?
The first year they were something like 30, 15 male and 15 females. The second year we made a larger amount, a larger plot lot of translocation, like 100 specimens. And now we are not translocating since last year, but we continue finding.
Was the area protected?
After we introduced the Zeuneriana, they decided to protect the area.
Okay.
About Pseudopruma baldensis.
Yes.
25 years ago, it was extremely abundant
Yes.
all over the Montebaldo region. And the last time I visited Montebaldo, I observed a very reduced population. Do you have
Well, I have been there last week, but it is too early now. But surely, Pseudopurna, it is one of these species that is decreasing. Okay. We are experiencing that for various species.
Bad news.
Bad news, yes. Thank you.
Okay, thank you. Carmela Adzik, I hope is correct. One season of Orthoptera research project in Croatia. Please.
Grazie. So yes, I will talk about research of Orthoptera in Croatia. Just yesterday during dinner, we were discussing how Orthoptera research in Croatia turned 10 years because Josip Skejo and Fran Rebrina started this modern period in 2013. Back then, it was very different than today. They didn't have any mentors. It wasn't really a recognized group. We didn't have enough data. And now it's different. We have much larger team. People like myself that joined the students, we were lucky enough that we had mentors. And I will talk to you about stuff that we were doing in 2023, sorry. So one of our projects was on Prianotropis hystrix, on eastern stone grasshopper. This project was funded by Mohammed bin Zayed Species Fund, Species Protection Fund. For this, we were researching Prianotropis hystrix. He is in this area outlined, it's not showing. Okay, in the area outlined in yellow. No. So for this, we were doing study on a number of populations. We were gathering data on botanical research. That was the part that Sebastian Chato, that is here, was doing. He was doing like a plant community on its type habitat. We are also collecting scat and leg samples to do analysis of population diversity, because we don't know how well connected these populations are, well, how isolated they are. And at this moment, we cannot really claim for sure that this is all one species. So this is something that we want to check. We want to check the taxonomy, make sure, so that we can know that our further data is accurate. And we're also doing specimen photography, where we are documenting their morphological diversity. As you can see on this photo, they can be very diverse. They can range from completely yellow to completely gray. They can have very different markings. So it is something that we are very interested in looking. If it correlates with their distribution or something else, we will see. This is still an ongoing research, so I cannot really give you any final data, but I can talk a bit about the stuff that we are still researching. So this species is locally abundant. This is actually something that is very well noticed also by locals. Prianotropis hystrix is only Pamphargida species in Croatia, and it is relatively large and yellow, so locals will recognize it. And when you say that you are doing on that species, some people will also comment and they will be like, oh, we remember it when we were little. They were much more abundant. One guy also told us that his grandma very much loved this species because geese, in the period where they are gaining fat, she was feeding them to these grasshoppers, and that was the way that they would get the most fat. So they were used in that way, but today we are probably seeing decreasing populations. We also have some historical records of local swarms. And today we don't really know trends because we lack historical data. So here we are doing like a checkpoint to have data to compare in the future. And also we have records in Croatia of children using them as toys. You can see why. They're very friendly species to work with. They live in a variety of habitats. Here you can see them on horse species. So these are two mating hystrix, and this is Orthoptera brevipenis, so they often share their habitats. And normally we find them in well-preserved habitats, and they seem to prefer a bit higher altitudes. They are threatened by many things, mostly by former grazing regimes abandonment, because this type of grazing where cattle are left to free roam is something that is decreasing in Croatia, and we are already seeing some effects of it, not only on Orthoptera but also on other groups. Here on the road you can see a lot of these dots. Now these are not all hystrix, but actually most of these are Orthoptera. This is a place where they are locally abundant on both sides of the road, and they cross it. You can see them when you drive, and sometimes when they're very active, you can't really avoid going over them. Our other project that is finished is research of Saga pedo. So this was a national project that was funded by European Union. Because Saga pedo is a strictly protected species in Croatia, and it is a Natura 2000 target species. So it is, this was a project for a lot of groups of animals. I think we had 20 groups, and one of them were Orthoptera, where we only worked on Saga pedo because it is the only eligible species for this research. All right. Okay. So this was conducted last year, and here you can see a variety of habitats where we did find Saga pedo, and maybe you can notice from these photos that these are all very well-preserved habitats. And in fact, we did not find Saga in any habitat that was facing some obvious human activity and some obvious larger threats. So we were sometimes philosophizing about Saga pedo and it being a protected species because it is not that rare in Croatia, it is relatively widespread. It is parthenogenetic species, so it shouldn't be that very endangered by nature. But during this study, we did notice that it is actually a great indicator for any threats that can be found in habitats because it is very sensitive. If there was any threat that we could recognize, those were habitats where we did not find Saga pedo. So research of this species is actually a part of a much larger context because data that we gather by researching this species can be also used and overlapped for many other animals and groups. Here you can see its confirmed distribution in Croatia. For these projects, we have, well, Croatia has these reporting periods, and they report in these 10 by 10 kilometer squares. So these outlined in red here are the ones that were confirmed in our previous reporting period, and the green ones are the ones that we added in this one. Now, these are not all our data, these are also literature data, but the point is that it was not available in the previous reporting period. And we have 3 biogeographical regions, though we could argue that we have 4, but that's a discussion for some other time. And in this reporting period, we added this, this entire region, this continental region. These are all the localities that we suggested for future research. I think this is for a period of following 24 years. So we suggested it to explore everything. We don't expect it to find on all of these places, but they should be checked. And also, we were, well, designing monitoring program for Saga pedo. And along some classical things that will be followed, we suggested that habitat succession should be a very important part of monitoring program. This is true for other groups, like I mentioned, there were 20 groups in total, and this is something that our ministry will work on, and they are developing ways to track this. So this we will have this in near future, probably. Overgrazing, because this was something that they were very sensitive to, we never found them on any overgrazing localities. And we also suggested complete Orthoptera diversity monitoring because, as published data also suggest, especially from Italy, Saga prefers habitats that have generally diverse Orthoptera fauna, and this correlates with our findings, so this is something that we suggested, and right now it is accepted, so we will see in the following years as it goes on. And this is when I show you those projects on map. So outlined in green is the research of Saga Pedo, where it took place. So it was mostly in this Mediterranean region, parts of Alpine, and this small locality in continental region. I must say that this locality was actually discovered thanks to citizen science. So it is something that is very important for us. And it is also great for charismatic species such as these because they are relatively easy to identify, and it is really a very valuable source of information for us. For Hystrix, these are localities that we will be looking at. And as you can see, most of this is located in the Mediterranean part. This is due to a number of reasons. First, maybe the biggest, these are charismatic species. So especially if you're applying for projects, if you are looking for funding, these are the species where it's easier to get. And we also have these endemic species, like Hystrix is mostly distributed in Croatia. It does have a bit of a range in some neighboring countries, but most of its population is in Croatia. So this is something where it's much easier to conduct research. And the other reason is that we have to start somewhere. So in the future, we are looking to extend our research to this continental part because in 2018, when a Croatian catalog of Orthoptera was published, islands and the parts of Mediterranean region were recognized as region where we lacked data. I do think that in the last few years we have contributed to that, so I think now our mountains and continental region is something where we would like to focus our future research. Apart from that, we are also working on Rachocleis buchichi. Here I am cheating a bit because this is a project that will be conducted this year, so 2024. Here is its distribution. And also some localities on mainland, which we will probably not visit this year, we will see. So its type locality is on Hvar Island. It is also found here on Vis, and we found populations on very, very isolated Palagruža Island and populations on Kornati Island. This is actually a national park. And these populations have strong morphological differences, and we believe these are actually 3 separate species, but we will see. Very important part of this is that National Park of Kornati recognized this, and they are collaborating with us this year. So we've come a long way because already Croatian institutions are recognizing importance of Orthoptera, and it will be something that we can look forward to in the future. And with that, I would like to thank you for your attention.
Thank you. Some question? Have you found some correlation between the presence of Saga Pedo and the calcareous formations?
We have not looked into it. We did find strong correlation of it preferring southern slopes, and it was white calcareous. Yeah, Kalnik is calcareous also. Yeah, that isolated population.
Okay, thank you.
So it should, yeah.
Thank you.
Thank you.
Antonia Donner, Grasshopper Summer School 2023 in Greece, inspiring the next generation of Orthoptera enthusiasts. Antonia? Che è successo? Non ho capito.
No, devo farmi dare perché non mi hanno mandato. Allora, we have
Emanuele
Emanuele
Repetto. Identifying ecological corridors of the bush cricket Sagapedo in fragmented semi-natural grasslands.
So I'm sorry, we will continue to talk about Sagapedo. So yeah, for the last 4 years, I conducted a study on the ecological of the Sagapedo, and now we present the results of these studies and also other things about that. So the distribution about Sagapedo, we talked about the Croatia before, and it's actually widespread in the Mediterranean area, but also near Asia, and it's very, very fragmented. We can see it's more concentrated in the Mediterranean basin, but also in other regions. Why so peculiar? Well, we already told it's parthenogenetic. It's also a predator, has a long dormant eggs, from 2 to 7 years dormant eggs, so a really long time. And it's also cryptic and elusive. Where does it live? Well, Carmela already told, but xerothermic grasslands, in particular some habitats from the Natura 2000 net, and natural or semi-natural, between 0 and 1,700 meters, so also with a wide range in altitude. So it's threatened by the expansion of the forest cover. It's one of the main threatenings for Sagapedo. It's also threatened by the intensification of agriculture because it's really sensitive to the change in management of the areas, and also from the abandonment of agropastoral activities, traditional, so extensive and not intensive. It's protected by the Natura 2000 net, and so the European Habitats Directive, but it's also present in the Berne Convention, and it's assessed as vulnerable by the IUCN. It has really low density population, so it's hard sometimes to find it, and it's probably under-detected, so it's probably the more present than what we know. It's elusive and cryptic. It's hard to find in the field. Also, if maybe you are not so used to search for saga, it's hard to find it. And it's often in relictive meadows, so in maybe not so reachable areas or just small meadows between the forests. And has a low detection probability. The studies in the left, it's our last studies. We estimated that the detection probability, you can see it's 21.01, so it's really low. For instance, some orthopterans can reach 70 or I think also 80% of detection probability. The aims of the studies are modeling the connectivity between the areas, identifying the potential ecological corridors, and suggesting where and how to intervene to maintain and maybe expand the population, because it's really fragmented and really threatened by the forest expansion. So this is the first study area. It's actually a small study area in between Piedmont and Ligury in Italy. You can see the 16 areas that we studied, and the borders in black are the borders of a Natura 2000 site. So almost half of the areas are inside a Natura 2000 site. The method: we conduct working transect of 10 meters long for 2 meters wide, and once per week between April and August, between the 2021 and then 2023, so the last year. In the 2022, we also surveyed the whole Orthoptera community to see the correlation with the abundance and the diversity of other Orthoptera. Because, of course, it's a predator, so see what it's useful. And here you can see the phenology of the species. So in cyan, we have the juveniles, and in red, the adults. On the x-axis, there is the session, so the period of the year. And you can see it's more easy to find it between the second week of May and the second week of June, because there are more juveniles, so it's more easy to find it. So here on the left, you have the sites divided in station, the station A, B, C, D, and the station of the B are all inside the Natura 2000 net. On the right, you see the abundances, and you can see that the station A, outside the Natura 2000 sites, has the 60% of the observation. And in this station, there are 3 areas, and only one area have more than half that all observation in all the 3 years. So almost all the population is only in one site. Then we collected the satellite data for some variable, and we use Copernicus and in Italy, so a source online for the satellite data. And we ran a variation inflection factor to see and avoid the multicollinearity. And we excluded all the variables that have more than 3 of variation inflection number. So we were left with 16 variables. And we have the shrublands, the grasslands, the bare spaces vegetated areas as percentage, but we have also the human population, the settlements, the standard deviation of the slope, and other that you can see in bold. So then we tried to do a species distribution models and use it to run the circuit theory, so to see where are the corridors and what are the best corridors for this species. We use, for sure, the variables from before to run the species distribution model. So we evaluated also the spatial autocorrelation between the eras. That's why we didn't use something like Maxent, but instead we use Omniscape, because we want also to evaluate the spatial autocorrelation and not exclude the data that are from nearby areas. So after the species distribution model, we made a map with the resistance to movements. Every point has a value between 1 and 0 that comes from 1 minus the probability of occurrence. So we have the resistance of each point to the expansion of Sagapedo. Then we ran on Omniscape, and we did the map on ecological corridors. So in the left, you can see the probability of occurrence. As you can see, more green means more probability of occurrence of the species. Then we covered it into the suitable areas between 0 and 1, and the red one are the suitable areas. They are the 5.57% of the whole areas. These areas is bigger than the areas of the study before. It's expanded by 25 kilometers in each direction, respect to the one before. Then these are some graphs about the probability of occurrence and the different variables. You can see shrublands, human settlements, distance to croplands. As you can see, some of them are pretty obvious. So more human settlements, less Sagapedo. It's pretty obvious. But others are not so clear, and we will check again and maybe implement some other studies to understand better some things, like the distance to crop lengths with this strange curve. Then the results. So on the left, you have the map of the landscape connectivity. So in yellow, there are the better corridors, the more suitable and the more expensive corridors. And in the right, you have the suitable areas in red on the connectivity. So you can see if two areas suitable for the species are connected and how they are connected and by how many ecological corridors. So what are the problems? What we have to do? We have more small fragmented areas. You can see in the red, it's really, really small areas and then nothing between one and another, except for some clutch. We have also limited ecological corridors. They are not that many, and they are also thin and not so suitable. So we have to intervene in the corridors, also with the deforestation, because as I told, the forest cover, it's one of the main problems with Sagapedo, also because it has a really low mobility, Like 10 meters of forest can be a barrier to Sagapedo. 10 meters of forest is nothing. So we have to intervene and try to maintain the connection between population, not obviously for the genetic flow, because it's parthenogenetic. There is no genetic flow, but because we need to maintain a high number of exemplars and maybe also expand it in other areas. That's it. So thank you.
Some question?
Okay.
Yes.
I have a question. You mentioned afforestation is a problem.
Yeah.
So what do you think? After the deforestation, after how many years Saga can recolonize?
That's a good question. That's a good question. I reverse the question. We had an areas that was before Amido, and then, like in 2023, it was reforested almost with hornbeam and ash woods. And we find some juveniles of Sagapedo, then after few weeks, went outside in a nearby meadow. So if there is the possibility to colonize a new areas, they do it. I don't know actually in how many years, but I think in 2 to 5 years, probably. Because also longer, yeah, for a stable population, yes, longer. But just for few exemplars and then the expansion, I think less.
One question. Why did you exclude Sicily from the distribution of Saga?
I did not. GBIF did.
No,
Yes.
because the data I'm not very sure.
Yeah.
Bodwin has a question.
Bodwin.
Yes. It's a difficult species to find, okay. Did you, we are experimenting with UV light.
Oh, yeah.
Working, and it seems that especially for Encifera, but maybe also for some other species, a good UV torch,
Yeah.
walking in the night might be a very good way to find these big specimens.
I never tried the UV light. I tried some night surveys last year,
Yeah.
but yeah, I think that it's a possibility to try the UV light.
In the dark, of course.
Yeah, but also there is a study by I think Anselmo from Val di Susa, and he used the UV light because he placed a target on the Saga, a target for monitoring the movements, A marker, yeah. And you can see in the picture that the saga is actually not so different from the environment, also with the UV light. So I don't know.
Okay.
But could be.
You really have to have a good UV light and not the cheap
Yeah,
ones.
yeah, that's
because
true. Yeah.
They are crap. You need a very good UV light.
Okay, now the coffee break. I recommend you to come back here within 15 minutes.
Before the coffee break, I want to remind you that is your last chance to buy books here from World Biodiversity Association. And we have the group photo downstairs in the garden, please. Summer school. Okay, just wait for the chairman.
Können wir das zur Seite schieben? Eins muss hier unten passen. That's absolutely okay. No problem. Vorsicht, Touch-Displays an. Tut mir leid. Sie ist heute irgendwie nicht so gut drauf. Ja, sind wir ja auch, gell? Jetzt irgendwie sind alle weg, kann das sein? Oder waren wir gar nicht mehr? Von hinten saß noch mehr.
Ja, von hinten saß ein bisschen mehr. Ich glaube, es fehlen nur noch. Ich finde es trotzdem irgendwie
Ja, schon.
vielleicht hätten wir doch noch mal.
Naja, nee, wir müssen nur zuerst bereit sein. Vor den anderen den Pointer ausprobieren wäre nicht so gut. Gute Wein, check das mal. Da ist unser Chato. Ist es an? Nein, ist nicht an.
Der wird uns ja auch noch ankündigen.
Okay. Now, Antonia Donner, Margarita Delles, Sophie Papczyk. Sorry. Grasshopper Summer School 2023 in Greece, inspiring the next generation of Orthoptera enthusiasts. Please.
Thank you. Thank you so much. First of all, thank you to Emanuele, who just took over when our presentation wasn't ready yet. And yeah, my name is Sophie Papczyk. I'm currently working as an ecologist for an environmental consultancy in Ireland. Antonia Donner, she's a master student of biology at the University of Cologne in Germany. And Margarita Delles, she finished her degree in microbiology and is now pursuing her second degree in pharmacy at the University of Bonn in Germany. We're really, really happy to be here today. This is our first conference, and we met each other last year at the summer school, and Luc was already mentioning it in his presentation this morning, and we now want to give you more details on the structure, on the activities, and from the perspective of the participants. So how did it all start? Luc, Roy, and Baudouin published their book, The Field Guide, The Grasshoppers of Greece, in 2018, and together with Vasily Kati and Axel Hochkirch, they decided to organize a summer school and to train a new generation of Orthoptera enthusiasts, and Greece was the perfect place for this because it has a high density of species and a high level of endemism and red-listed species. They found support in several institutions like the Naturalis Biodiversity Center at the University of Ioannina, the city of Konica, the Feriaki Fund, and UNESCO Geopark in Greece, and they supported the whole event financially. We got free accommodation in a student hostel of Konica, and we also had lecture hall and laboratory facilities. Again, at this place, a big thank you to all of you for organizing this, and also for the support we received from Eleftheria Zacharopoulou, who was in charge of the healthcare. Also, we had several guest lecturers from different countries. There was Lisbeth Zechner, Konstantina Sografou, and Haritakis Papayanou, who were having presentations, both in online lectures and on site. We were a group of 20 participants coming from 7 different European countries, and the most of the participants were from Greece and from Germany. It was a really good mix of experienced and beginner people, so we could interchange our experiences, like some people were already writing or working on a PhD and others were just beginning to dive into the topic and the lovely group of grasshoppers. And now Margarita will tell you more about the activities during the summer school.
So, 2 weeks before the start of the Orthoptera summer school, we had 4 online lectures. In these, we received an introduction to grasshopper ecology, systematics, field techniques, research, and conservation. And we could hardly wait to meet each other offline. The venue was in Konica in the Epirus region of Greece, near the Aos River. This location is known for the ancient stone bridge. Konica is located about 5 kilometers south to the Albanian border, at the point where the Aos River exits its valley, between the Smolikas Massif to the north and the Tymphi Massif to the south. We were very fortunate that everything was available at the venue: the accommodations, the food facilities, the educational center for lectures, and the laboratory. Thanks to this great setup, we were able to spend every single minute of the summer school together. This is what a typical day during the summer school looked like. In the morning, we had breakfast together, followed by field excursions. After lunch, we had lectures and lab work. After dinner, there were night excursions for the hardcore enthusiasts. These usually ended between midnight and 3 a.m. in the morning with a beer, so we didn't get that much sleep. During the field work, we practiced the applications of various methods, capturing with nets and subsequent identification using the field guide, evaluation of the catch from 5 by 5 meter quadrats, behavioral analysis and acoustic detection at night. And the behavioral analysis was, for example, really funny for us because, like you see in this picture, we were staring at one individual grasshopper for 10 minutes and observing if it was eating or stridulating or mating or, like in our cases, mostly resting. As mentioned, we also had night excursions. We were introduced to the technology of bioacoustic and were able to practice handling the bat detectors. We also saw many other species and enjoyed the local biodiversity. We had 4 excursions in the Konica region. The first excursion went to the hill slopes around Konica. The second went to Mount Gramos. The third went to Baba Hill. And the last one to Caravina Lake. As you can see from the photos, these were very different habitats. For example, the hill slopes around Konica and the agricultural school are abandoned agricultural land. The Gramos region is subalpine grassland. The Baba Hill is mountainous forest and grassland. And the Caravina Lake is wet grassland.
At the venue, we heard several more interesting lectures about the local UNESCO geopark and field photography and bioacoustic techniques and the conservation of grasshopper species. The afternoons were used to do some lab work. There we identified the specimens that could not be identified in the field with the help of microscopes and identification keys. And the teachers also showed us how to prepare the specimen. And we documented all the data that we collected in the field. In total, we found 50 different species during the week. And here you can see some of the examples. All these photos, like the other photos we show you during the presentation, are taken by participants of the course. First, here we have Saga helenica, which was a huge highlight for everyone because they're one of the largest species in Europe, so we were really happy to find them on Baba Hill. Next to it, we have Decticus albifrons, which was very commonly found around the area of the venue, and especially at the night excursions, we found a lot of them. Here you can see one species of Psilimon that we found. This genus is a very species-rich genus in Greece with 46 species. In the bottom, you can see Conocephalus kisi, which has a very long ovipositor. And next to it, another highlight, Paracoloptenus caloptenoides, which was another highlight, like I said. because it is protected by the European Habitat Directive. And lastly, Oedipoda caerulescens with its beautiful blue hind wings. At the last day, we took an exam and we did group presentations on one species that was found, each group. And in the evening, we went for a nice dinner and got our certificates handed out. The summer school really inspired us because we could discover some of the amazing grasshopper fauna of Greece and develop a sense of responsibility for nature conservation. On top of that, we had a wonderful time and made lots of new friends, and everyone passed the exam and obtained the credit points. So all in all, the summer school was a chirping success, and we want to give a huge thank you to all the organizers, teachers, and sponsors, and we really hope that events like this will happen more often to train the taxonomy experts of the future.
Thank you very much.
Grazie.
Some question? Yes, Axel.
Not a question, just a remark. Something you have skipped. That was, in fact, the hottest week in Greece last year.
Yeah,
That's
it
true.
was really hot.
It was a heat wave.
But we still, so we are also happy that the participants all survived.
Okay, thank you. Compliments.
There's another question.
Ah, yes, sorry.
I was wondering if that being organized every year somewhere, we did the first time at this conference. And if not, I think it would be very great if that could be just organized in different places all over.
It should.
Yeah, definitely.
What I can say is that next week we have a workshop that is focused on bioacoustic in the Museum of Verona. It will be repeated in Spain in August and in Greece next year with both.
I think it's just a very great example as it was organized there in Greece, and maybe other countries may organize a similar workshop.
Yeah, we wish the same, yeah.
Are you planning this year to organize also? We want to ask you to organize. I could ask the faculty because we have a building in Dinari College which can host
Okay. Thank you. Roberto Battiston, the GPT Red List. Can artificial intelligence help in species conservation?
Hello, everyone. I have to warn you that this will be a pretty strange, weird presentation, but I'm trying to give some food for your thoughts and for your curiosity on this new technology which is entering in our lives, even if you like it or not, and how to apply this in our work for Orthoptera conservation. Okay, the new generation of artificial intelligences, large language models are now in our society, even if you like it or not. And they are entering also in the science, in the field of science. There are clearly some positive impacts and some negative impacts of this new technology. I'm not going to list all of them, I'm just trying to give you some examples of positive impacts and negative impacts. It is a very high energy demanding technology, can generate electronic wastage, and is creating some ethical scientific problems. Maybe you know that there are many scientific articles written by ChatGPT, sometimes cited as co-author, sometimes not. There are a huge number of questions that is rising up in these recent years. Maybe you have experimented this technology in species recognition. Maybe you have tried the iNaturalist algorithm for species identification, which in the last few years was complete junk. But in the last year, it is making amazing progress. You can identify, at least at genus level, pretty much everything. There is a good application also in sound identification, but we'll talk about this tomorrow, I think, so I'm not going deep in this. But there are some very, very interesting applications for identification, species, sound, or so on. I'm giving you 3 possible applications for species and Orthoptera conservation. An assistant to our work. Some help in statistics for advanced population trends modeling, and something on education and communication awareness. So I created as an example a species conservation assistant. You know, you can try it online. It is online. You can try with this QR code, or you can find it in the list of the GPTs of OpenAI. You know that OpenAI, the creator of ChatGPT, has created also I've opened the possibility to create GPTs, chatbot that can be trained on specific topics, and they can be a very useful assistant. This assistant is made to assist you in the work of conservation in a general level. Can help you in calculating the extent of occurrence of a species, to draw maps, to give you the interpretation of some particular situation on some species, interaction with other species. It's like a colleague, an old colleague, where you can chat on some very deep topics. It is a chatbot, so you can chat like your colleague. Maybe chatting with your colleague or your friend is nicer, but maybe he doesn't know anything. This chatbot is has a large knowledge. This one has been trained with all existing IUCN literature for conservation. All the guides and manuals that are present in IUCN website are in his knowledge. So you can ask him anything about, for example, calculating the extent of occurrence, recalling particular procedures, notes, aspects of the laws, the conservation, interpreting the data you have from a field research, for example, in the light of conservation. And I think it is a nice application is to train students. Training for complex procedures can take time, can take persons to train other persons. So this is a teacher, a virtual teacher you can ask to do the basic work. The second application is about population trends modeling, which is, if any one of you have tried to do a red list of a species, having a population trend is a dream, is a unicorn. Here you can see a grasshopper unicorn. Global population trends are very difficult to obtain from field records, are a dream for everyone who deals with red lists and conservation. The modern AI models offer a powerful language interface to discuss complicated statistical data, even for non-statisticians. Okay, imagine that you may have the starting set of data for population models, but you are not a statistician. Maybe I'm a taxonomist, maybe you are a taxonomist, ecologist. Working with high-level statistics is very hard for me and maybe for you. You need a mathematician, a statistician. Well, this is a colleague we can discuss on which is the best model, best statistical model I can use for this particular situation, which is a very good help. And it is a language interface. It's not numbers, formulas. It is a chat. In your opinion, which is the best way I can deal with this problem? And the answers are often very, very interesting. I made a test, an experiment of this application. I asked which model is best, and I tried the random forest model, which is a machine learning algorithm model. Humans cannot do this, only a machine can do this statistical model, to predict the population trend of all the European Mantodea species in the next 10 years, which is a dream for me that I work on mantis, and I want to know if they will be present or not in my red list in the next years. Now we more or less have the starting set of data. In a naturalist, there are a lot of verified good records for single present records. I used a lot of them, 50,000 verified records, and I asked my assistant to calculate the extent of occurrence for each mantis species present in Europe in each of the last 10 years with a single prompt. Prompt is a command line, which is not maybe a line, but a paragraph, but it is a very simple question to ask to the machine. It was able to manage all this data. Of course, all this data was weighted because the iNaturalist platform is increasing each year. If you have many more data each year, doesn't mean that the species is increasing. Maybe the observers are increasing. So I used some filters for this data. And then I asked to apply this random forest model to this data, to the data for the last 10 years, and predict the next 10 years. I normalized the result and plotted them in this beautiful picture. Here you can see on the left, these are all the species of mantids present in Europe, and all their present records. In the left part, there are the past 10 years, with, of course, many gaps, because we have many data, but not all of them. And in the right part, there is the prediction. It is something amazing to see. It's the first time I think I see something like this. But the big question is this a good prediction? Is this result good? The short answer is no. As a mantid specialist, I don't trust very much this result. I see something very good and something very bad. You can see there is a big difference from the left part and the right part. So maybe for something the result is good, but for some others it is not good. Maybe I'm wrong and he's right, but I feel that there's something not working very good in this. But the long answer is that, to me, the result is good, because the model is very simple. I use a few hours of modeling to get this result. And in artificial intelligence, the starting point is the core. If the prompt is good, and if the starting set of data is good, the output will be good. If the starting point is bad, the result will be bad. The human part in this process is still big and have a huge impact. So this model can be improved, of course. And I think that can be the starting point of a new way of modeling population trends and every other aspect of ecology and big data of species conservation. I think that it is an half open window because it is an experimental window on the future of population trend modeling and species conservation. So take these, and also I have to say that you must be very careful when you ask calculation to a language model like this. They are made for chatting. not to make calculations. So you have to double check, verify the result, and do not trust them at the first time. But artificial intelligences are made of algorithms, mathematics. So if you scratch the surface, you can emerge the real core of this powerful technology. But you can do your best scientific research, and if you cannot communicate it, it can stay in your office drawer and be not very useful. We scientists need to find ways to communicate our science, our result, to have an impact not only on scientists, of course, but on maybe politicians, maybe citizens, the person who have a big weight on species conservation. And especially the new generation, we have to leave something to the people that will come after us. But doing conservation and communication awareness requires a lot of resources, time, maybe to have a multi-target audience, a new generation, politicians, lawyers, and many different targets that need different languages, different communication plans. It's very complicated. And maybe it's not our job. We are scientists. We need to hire somebody, some communication specialist, to do this kind of job. But maybe we don't have the money for this. And there is an assistant to do this kind of job we can use, almost free. I tried to make an experiment. I asked the Suno AI, which is an artificial intelligence made for music, to write a song of a conference of Orthoptera conservationists, where the conservationists, us, are superheroes fighting to protect the planet, because we are cool. And we need to communicate that species conservation of nature is something cool, that we need to, it's not something for all scientists, but it's something very cool, like superheroes. How do you communicate this to young students? I asked this artificial intelligence to create a song to sing, play, and make everything we need to have a song for take this as a celebration of our work and also of this meeting. It's something very strange. But I have not hired a rock band, but everything is made by an artificial intelligence. The result is pretty good.
In a dim-lit room where the heroes gather, all Orthoptera saviors, ready to fight and to matter. Crickets, grasshoppers, mantids by their side. Insect defenders make passion their abide. They speak of extinction, tragedy to see. What's their mind? Not realizing what it means. The signs of summer nights, we got the songs of the storm. They won't rest until every insect is reborn.
Okay, this is imagine this on Spotify or in some communication campaign on the social media. It is very cool. And okay, but the problem is that I use rock style music because I suppose that this audience was more or less of my generation. If you want to involve teenagers, they don't care about rock and roll, of course. They want trap music. I personally don't like trap music. I have no idea how to use this. But I asked the Suno AI to convert this rock music in trap music. This is the result. Oh. Oh.
In a dim room where the heroes gather, Orthoptera saviors ready to fight and to matter. Crickets, grasshoppers, mantids by their side. Insect defenders with passion they'll abide. They speak of extinction, a tragedy they've seen. The world's turning blind, not realizing what it means. The silence of summer nights without the songs of the
okay, maybe you don't like this, but teenagers will love this. So, in conclusion, let's play together. AI is opening a lot of new opportunities and threats for science. It is almost an explored field with many things to try and to experiment. I have a proposal for you. Maybe some of you is already playing with this technology. And my proposal is to set up a working group on the application of artificial intelligence on conservation. Maybe inside the Grasshopper specialist group or some conservation group. There is my email in your booklet of abstracts, so feel free to write me, to contact Filippo or the organizers. And I think that we need to join our forces and play together because there is a lot to do. There are not many people working on this, but there are huge implications in bad and good ways of this new technology. Are you in? I hope so. And I leave you with a fake citation speech of a fake conservationist just to make you think about the future. Thank you very much.
Have you some question? Soddisfatti. Ok. Thank you. Dove siamo? Ah, ecco. Sì. Philip
Kirchner.
Glacial Legacies, Refugial Dynamics of the Endemic Bush Cricket, Anonconotus Italo-Austriacus, again.
Again, yes. So thanks for the introduction. Thanks for inviting me. I'm the second Filippo talking about Italo-Austriacus. And I will talk more about the evolution of the species. Thank you, Roberto, for waking up the audience at this time of the day. I hope that you enjoy my talk. So I'm kind of a hybrid. So right now I'm working at the University of Innsbruck in botany, but I had previous projects on grasshopper evolution together with my colleague Petra Kranebitter, who you can see on the right and sitting in the back. So first of all, the genus Anonconotus, it's a perfect model species to study evolution of an alpine species, of course. It's endemic to the Alps, and where is the mouse? It's here. So what you can see here, it's endemic to the Alps and the, oh, no, it's not working? Ah, it's working. So this is the distribution of the whole genus. There is several species all over the Alps, and one species in the Apennine. The species is large and flightless, which it gives it a limited dispersal capacity. It occurs exclusively or almost exclusively above the tree line, so it's a true alpine species. And in my experience, all species, they inhabit alpine meadows and these open dwarf shrub habitats that Filippo showed you before. They have this interesting coercive mating system that Karim Vahed studied intensively, which I cooperate, with whom I also cooperate on this project. And most interesting for me as a biogeographer is that the range of the species, all of them, it's fragmented and sometimes it's also very small. Today I will only talk about Anonconotus Italo-Austriacus, so this is a phylogeny of the whole genus from a paper that I hope to publish soon. Let's see. So all of the criteria that I mentioned on the previous slide also apply to Anonconotus Italo-Austriacus, of course. This species is endemic to the Dolomites and to the Hohe range, which is a mountain range shared by Italy and Austria. Whereas, so it's listed as endangered in the IUCN assessment. And as you can see here, plotted on this map, all these tiny dark spots, I don't know if you can see it, so it's all known occurrences from GBIF together and my own observation. So it's very island-like in the south. In the Dolomites, it's very fragmented. And in the Hohe Tauern range, it's a bit more continuous, the distribution. So the gray spots that you can see here, these are all the populations that we have sampled. Forget about these R's that you can see here. I will mention this later again. So large parts, or I would say all of its habitat was covered by glaciers during the last ice age, which makes it interesting to study the refugial dynamics also of an alpine species. So this would be the black line. It's the outline of the last glacial maximum. Oh, it's not working. So just to give you an idea, because I will talk a lot about refugial ecology too, the two most important types of refugia for alpine species. It's not working. Nope. Yes. So the two most important refugia type for alpine species are either nunatak refugia, which are peaks or ranges of mountains or groups of mountains that are protruding the ice shield but are surrounded by ice. This is widely shown for plant species, and it's also shown for some larger arthropod species, for example, for these Trechus beetles or also for Machilis. But more important, and probably the most important refugial areas for larger insect and arthropod species are, of course, peripheral refugia, which are foothills, unglaciated foothills of mountains, which were present on the southern margin or the eastern margin of the Alps. So why are we interested? Oh, there is something cut off. So why are we interested in refugial dynamics in the first place? Because they're essential to understand the evolution of an alpine species. And second, what has been cut off, they're also important for the conservation of the species. Very often these are areas where genetic diversity is really elevated, so where we find the highest levels of genetic diversity within a species. So the hypothesis that we had, so this is also shifted, unfortunately. The hypothesis that we had on the refugial, so we had three hypotheses on where were the refugia for Anaconodus italo-austriacus. So the first was that there was one pure southern refugium close to this present-day population of the Vettefeltrine, and all the other populations were colonized. All the other places were colonized from there post-glacially. The second hypothesis, there were several of these refugial areas, maybe on the southern margin of the ice shield, or some of them on the eastern margin of the ice shield. And last, oh, it's not working. Nope. Ah, there it was. So the last hypothesis is, in my opinion, it's not very good anyway, but we were considering it because colleagues from Padua suggested it. It's in situ survival on nunataks. So all of the populations that we see today really survived on mountain peaks protruding the ice shield. So how did we evaluate, or how did we test between these hypotheses? So first of all, we sampled 36 individuals from 6 populations, more or less from the entire range. Would have been nice to have a bit more, but it's representing the entire range at least. We extracted the DNA. We did some fancy sequencing, so we did some genotyping by sequencing, resulting in several million fragments per individual, per population. And we had a look at genetic structure, distribution of genetic variation. We did some demographic modeling, and we also constructed some time-calibrated phylogenies to see when all of this happened. So this is the first result on the genetic structures or the distribution of genetic variation within the species. And what was clear to us was there is 3 huge groups. So first of all, we have a group in the Vettefeltrine, which is quite divergent from the rest. Then we have a very divergent group also on the eastern margin of the distribution, and everything in between here, it's not very derived, and it's very homogeneous and probably very recent. We found evidence for, so this is this red arrow here, evidence for genetic exchange between these huge inner alpine populations in the Dolomites and the eastern populations, but no connection whatsoever to the south. So this Vettefeltrine population is obviously very isolated. And we found the largest private allelic richness. This is some proxy for genetic diversity on the southern margin in this refugial, or in this potential refugial area on the south and on the east. Whereas in the middle, the genetic diversity was quite low. So it's not working again. No. And something else that was interesting is we found some weird genetic signal within these central populations and also a little bit in this southern population, which could in fact be like some old signal from this population from Montebaldo that has been introgressed in the past. But it can also be just some trash. So would be interesting if one day we will find it again, we could test this. So this is more a descriptive analysis now, but we were also interested in the processes shaping these patterns, and therefore we employed some explicit demographic model testing. I will not go into detail here. What we do more or less is we present like a huge amount of different scenarios of splitting and gene flow possibilities, and in the end we will run an algorithm, and the best model describing what has happened, or which is closest to what has really happened, is selected. And what we found here was, of course, that it's not working. This population in the south was derived first, so this would be here, and the 2 other populations split later, and that was some ancestral gene flow between those and all the rest, and then there was some unidirectional gene flow into the central population. We had a closer look also. No? It's not working. Yeah. Sleeping.
He will solve it. The guy will solve it.
So the computer is tired already. Oh.
Do you have any questions? No.
We could listen to the song again in between.
Maybe the artificial intelligence can take the control.
Yes.
But I suggest jazz style.
Yes. Oof.
But I can say it is very interesting because your view is wider than what I am doing now, because you have this possibility to check all the populations of the species, and this will give a very a larger view. I hope that in the future it will be appreciated and will give more results, reliable.
I think what is really nice is that importance of this Vettefeltrine population, it's
yes.
clear because it's very isolated. It's
Yes.
now.
Okay.
Okay, so
Let's try.
yes. No. Yes.
No.
Woo, it's working. So, as I said before, so we did some complicated modeling. What we found was that this population in the south was split earliest, and that the other two are more or less very closely connected. We also had a look only to what happened between this eastern population and the central one, so these Dolomite populations. And obviously, there has been clearly a founder event. So the three best models that we found, they were all more or less similarly likely, showed us that there has been a founder event from this eastern population into the Dolomites. So there was no recent contact of the southern populations with any of the other populations. Yes, so it's quite isolated. What is the time frame of all of this? So we had a look at the time-calibrated phylogeny. So we calibrated this in two independent ways. And what we found was, first of all, the split from the sister species, Anoconotus alpinus, that is occurring more north of the Alps. It's occurred about 1.8 million years ago, so it's early to mid-Pleistocene period. And that the Vettefeltrine populations, they have been more or less isolated since at least 220,000 years. All the rest, especially the center populations, emerged rather recently. So especially these green populations, they emerged quite late in the last glacial period, possibly even post-glacially for some of these Dolomite populations. So what we can say, the populations in the periphery, they are older, and the populations in the center, they are much younger. Whoop. So in situ survival, or nunatak survival, or old lineage age, that's something that the colleagues from Padua came up with. They suggested really old age up to 2.2 million years. We don't think that it's possible for these species to survive on nunataks, even if they suppose so in the discussion of their paper. And also their findings are not reproducible, and this is probably to methodological shortcomings, and our evidence clearly contrasts these claims. So we believe that the survival of the species occurred in peripheral refugia, and we think that at least two refugia were involved, so one in the south and one in the east. So more or less everything was colonized from the east to the west, and the southern one was isolated. So and finally, we have a possible evolutionary scenario, also together with this time-calibrated phylogeny that we present, that might fit also glacial stages. So first of all, 220,000 years ago, we don't really know how the Alps looked like, but there was, of course, some glaciation going on. So populations were still together. So probably they distributed along the southern margin. 100,000 years at the end of the last interglacial, there might have been some glacial expansion, and these lineages, in this case the red and the green ones, might have been separated. During glacial stages, 20,000 years ago, probably there were 3 different refugial areas, and that's how it looks like today. This is a bit hypothetical still, but the glacier models, they are accurate. They are from Swiss geologists. So maybe it's just coincidence, but this could be some hypothesis. So what we suggest, that there was an east-west expansion along the southern margin of the Alps, probably starting in the penultimate glacial period. So some take-home message: there are 3 distinct genetic groups. There were multiple refugia involved in the diversification of the species on the southern, eastern, and maybe even southeastern margin. All the inner alpine populations, they are young or of late or post last glacial maximum origin. All the recolonization occurred from the east, but not from the south, and there is no evidence for in situ survival. And populations of Vertebratrine, they are special, they persisted for a long time there. So thank you for your attention, and maybe there is time for some questions still.
Please.
So these are my collaborators. Thanks to all of them too.
Thanks, Felix, for your talk. Really like it.
Thank you.
How did you calibrate the
Sorry?
How did you calibrate the tree?
So we calibrated the tree using, so I got data, like mitochondrial data from Scotland, and I used the mitochondrial data that was generated in Padua, so independently, and then I created some consensus, and that was, so I did these calibrated ones, and I also ran some with just a mutation rate that more or less showed similar results, but the uncertainties were much larger. But both of them retrieved like young ages.
Axel?
Yes, you said that the note said that there was a bottleneck during the colonization from the red to the green. So are the green populations genetically less diverse than the red one?
Yes, yes,
And
clearly.
the yellow is probably the genetically diverse.
Yes, the yellow has the most elevated allelic richness, yeah.
Paolo?
All compliments for the extraordinary work of the group that worked at this
Thank you.
very lovely topic. And I imagine that it would be extremely interesting to do the same kind of analysis for other endemic species in the Alps. I can imagine Pseudoclima valdensis, Cotopodisma covelli, or other alpine species in general, very isolated, because of course during these glacial ages happened to Anoconopus, happened to the other
Yes,
species.
yes,
It's
yes.
difficult to imagine that another Orthoptera survived in a different way.
No.
So I think that the doubt about the precision of the calibration of the time can be enforced strongly by the overlapping of other studies. And I think that this kind of study will be part of the school for many orthopterologists.
Thank you. Thank you.
Very much.
There are, other than genetic difference, also some morphological difference.
I think you have to ask the colleagues from Italy.
I tried to start a study on the Vertebratrine
No, among all the populations, non inside the one population.
we started with Antonio Galvagni to study Anoconotus italo-austriacus, and we searched for the characters, first of all, to distinguish the species. And it's nice to see that genetical data support most of the species. I was very surprised that Anoconotus mercanturi was like an old parent.
To all others.
Yes, and it has some peculiar characteristics, but probably studying morphological, it will be possible to find something, but we have to explore this way too. But genetic data are still very strong.
Okay. Thank you.
Thank you.
Giacomo Ortis. Biological relation between the red listed species Barbitistes vicentinus and a ground-searching parasitoid.
Okay, let's talk about Barbitistes vicentinus. Okay, Barbitistes vicentinus is an endemic bush cricket, endemic of the northeast Italy. It can be found in the Bari-Ciongania hills, in the Lessina mountains, and it was found in a valley of Trentino region. It was described very early in 1993 by Galvagni and Fontana. And because for its small distributional range, it was considered as an early treated species in the IUCN Red List. So the species was considered rare until 2008, when the first outbreaks occurred in its southern range, that are the Euganian Hills, firstly in some local areas, but in the subsequent years affecting all the hillside area, causing severe damages to broadleaf plants as well as crops and such as vineyards and olive groves. So let's have a focus on its life cycle. So the eggs are laid in the ground of forest areas and can remain in the soil for several years in a state that is called initial diapause, where no embryonic development occur. Otherwise, the eggs can develop in the same year of oviposition, reaching a stage that is called the final diapause stage. So as you can see, the embryo occupies the whole egg space, and there is a green coloration. And after reaching this stage, the egg will hatch in the next spring, and then we reach the adult stage around May. So basically, in the ground you can find different types of eggs and of different ages. So during surveys in the Euganian Hills to monitor the density of this bush cricket, we extracted the eggs in the ground of 40 sites in the Euganian Hills, and we found some eggs with small circular holes in the corium, as you can see in the bottom left, and we tried to reconstruct the presence of these eggs, and they were present in almost all the sites in the Euganian Hills. In addition, because we don't know what eggs come from, we found some eggs with small circular embryos inside, and after incubating them in a laboratory, we found hatching a lot of parasitoids that are mimariid parasitoids. We found that from one single egg can hatch up to 136 individuals. So given this information, we carry out, we want to do more about this parasitoid, because the number of individuals that can hatch from a single egg was very high. So we want to know the real field parasitization rates, then the emergence period in nature, and we carry out also a laboratory trial on egg age preference, and also we carried out a population genetic analysis of the subpopulation in the Euganian Hills. So how we do it? Basically, we go again in the Euganian Hills, we choose 5 outbreak sites, and we extract the egg from soil from a surface of 2.25 square meters. And after extracting the parasitized eggs, we placed them in a laboratory and till the emergence of the adults. When we had the adults, then we carry out a laboratory trial on the egg preference of the parasitoid. We carried also the genetic analysis on the adults, as well as we counted also the sex ratio. And also with a batch of eggs, we valued the emergence period in a natural condition. So the laboratory trial on egg age preference, basically we place in one little vial one female parasitoid to 2 host eggs of 2 different ages. So to do this, we performed 2 different rearing of Barbitistes during 2 different years, one in 2020 and another in 2021. So in this vial with the parasitoid, we placed the 1-year-old egg, that is 1 egg in initial diapause, so an egg that didn't develop, but an egg that didn't develop and it was 1 year old. So we placed one of these eggs and 1 newly laid egg that come from the rearings of 2021. And we replicated this experiment 91 times. In addition, we perform a genetic analysis of the subpopulation of this parasitoid in the Ghanaian hills and prelevating 1 female parasitoid from at least 6 different eggs for each site. And then we amplify 2 DNA fragments, 1 nuclear fragment and 1 mitochondrial fragment. So results. As you can see, as I said, we prelevated the eggs from a surface of 2.25 square meters, and the egg of Barbitistes vicentinus were present in egg number like 500 eggs for every 2.5 square meter. However, we found low parasitization rates, around 2 or 3% for each site. And after the incubation of the parasitized eggs, we found that the sex ratio was strongly female biased, and we counted an average of 95 individuals hatching from each egg. So in total, we were able to collect at least 30 eggs. Regarding the laboratory trial on the egg age preference, we found that this parasitoid was able to parasitize on both egg categories, so we're able to parasitize on 1-year-old egg as well as a newly laid egg. However, we found a significant preference for the old eggs of the parasitoids. So the parasitoids prefer to reproduce better in the old eggs compared to the newly laid eggs. In addition, because we have also a control group of eggs not exposed to parasitoid and 1 group of eggs exposed to parasitoid, we found also that the exposed group of eggs contained a higher number of unviable eggs. And we related this effect to the oviposition of the parasitoid that was able to cause host egg abortion. Regarding the emergent period, we found the emergence occurred from the last week of July to mid-August, when the adults of Barbitistes vicentinus are almost died. But this period coincides also with the laying period of other tetygonids. So maybe, probably this parasitoid could be also a generalist parasitoid. However, we didn't found it hatching from eggs of other tetygonids in our surveys. Regarding the genetic analysis, we found 4 total haplotypes among the sampled sites, with 2 main haplotypes diffused across almost sites, and the presence of shared haplotypes and the wide distribution of this parasitoid in the area suggests a natural dispersal ability of this parasitoid that was able to form a stable population characterized by a low genetic structure. In conclusion, we found low parasitization rates that could be due to a possible time lag in the parasitoid host system. So because the outbreaks of Barbitistes start to occur 10 years ago, the parasitoid can't have time to reproduce at the same extent. And also, because the increasing summer temperatures have shortened the period of prolonged diapause of Barbitistes, decreasing the number of old eggs in the ground that are preferred to the parasitoid. Both these facts can have contributed to the low parasitization rates that we have found. However, there are three factors that can contribute to suggest a potential impact of this parasitoid on Barbitistes populations. That are the high number of individuals that can hatch from a single egg, that are around 100, the possibility to cause the host egg abortion, and the high natural dispersal ability and the wide presence in the area. It will be worth in the future years to monitor the density of the bush cricket as well of the parasitoid to better understand the dynamics of these two species. Thank you.
Axel.
Would like to know how easy is it to find the eggs in nature, the eggs of Barbitistes?
Okay. Because for this species was pretty easy because during the outbreaks it laid only in the forest ground, and if you go around casually you can find eggs. I go like around casually and I found eggs. But I know that for other species not to be the same because I found also the eggs of Orthoptera, Tetigonia, Orthoptera, but just one or two.
Yes.
You mentioned there the sex ratio in hatched parasitoids, 92 females, percent of females, and 8% of males. How do you explain it? Or is it a general pattern within the parasitoids?
Yeah, it occurs some pretty. See, it's a general thing in the parasite. General ratio in the parasitoids that they produce more females than males. Yeah.
And is known the parasitoid to parasite adult Tigonide?
No, because there are no.
Is it specific?
Maybe, but we don't know because this parasitoid occurs also in other parts of Europe, like Romania, also Sweden. So maybe it can parasitize also other species.
Ah, yes, yes, yes. Paul.
The biology of this parasitoid is not known, so only the species is known.
Yeah, it's not only the species, but the biology is completely unknown. See, there are only records in some parts of Europe that people found it in the yellow traps and stop.
Okay. Okay. Thank you. Mattia Ragazzini. Where is? Ah, here. DNA barcoding and the species delimitation of Orthoptera from the Mediterranean basin. Please.
Thank you. Well, good afternoon, everyone. My name is Mattia Ragazzini. I'm a research associate from the University of Bologna, Italy. And today I'm going to talk to you about DNA barcoding and species delimitation performances on Orthopterans mainly from the Mediterranean basin. Well, at first, I am quite sure you have already heard about DNA barcoding and its benefits and usage, but here a very little introduction. Well, the DNA barcoding usage is based on the application of a standardized sequence of DNA, of mitochondrial DNA, called, in the case of metazoans, it is the cytochrome C oxidase 1, subunit 1, also called COX-1, and but for other organisms, of course, need the use of other markers. And it's a method useful for the species level identification of unknown biological material. Its main benefits are the usage of small DNA fragments, but also the theoretical minor variation, genetic variation within the species, the same species. And, but it's also useful because you can easily amplify and sequence this sequencing thanks, for example, to conserve the flanking sites, which simplify, for example, the PCR process and the design of primers for the amplification. But here, I introduced the project I performed during my Master of Science thesis project concerning the Mediterranean Orthopterans barcoding, where we assessed the several species of Orthopteran from all Southern Europe, including Spain, Greece, Italy, France, Portugal, as you can see here, which are representative of a great Orthopteran biodiversity, but also 15 more countries. So we had an extensive data sets. Here you can see a map with the main, with all the sampling localities we assessed. The initial data set we got included almost 1,500 newly generated barcodes from 275 recognized species with a great number of unidentified records. But before performing further analysis, we decided to integrate this initial dataset with two more datasets. One concerning mainly the barcodes from Portugal, the EBOR project, which is still unpublished, while we also integrated the ZBORD project from mainly including barcodes from Germany, Austria, and Switzerland. So we got a final dataset of 2,630 barcodes from 20 European countries. Then we inferred at first a maximum likelihood tree for both caeliferans and ensiferans. As you can, I have to precise that we performed the subsequent analysis in a different way for caeliferans and ensiferans, in a separate way. As you can observe, the maximum likelihood tree show the monophily of most of the main represented families and subfamilies, except for some exception. But at the same time, it's important to say that DNA barcoding is not suitable for the deep phylogeny of organisms, but it's very, or possibly useful for the species identification and delimitation, as we will see. So to assess the performances of the species of the barcoding for the species delimitation, I adopted different delimitation methods based on different theoretical frameworks. Here some the methods I used. The two main categories concern the parameters used for inferring the molecular operational taxonomics units, which are the most likely species cluster inferred by these methods. So the first three methods, A, B, C, D, ASAP, and BIN, were based on the genetic distances among the sequences which were involved, while this clustering-based approaches, ZMC and PTP, involved the inference of a phylogenetic tree, ultrametric or non-ultrametric. Then I summarized the result using a new tool called SPDOL from Ramirez 2023, which were able to where is the pointer? Which we were able to represent the delimited clusters with a black bars you can observe here, one for each the combination of parameters and each method, while the red bars represent the consensus among the consensus of at least 50% of the methods involved, while all the specimens are listed on the right. So I got of course, a huge list of specimens and clusters. While on the left, you can observe the inferred species tree. Well, here an overview of the Orthoptera biodiversity coverage I was able to obtain. So we were able to cover all the biodiversity of Orthopterans from Germany, but also a great percentage of biodiversity of Portugal, and also a good result for Spain and Italy. While Greece, we still have a lot of work to do, actually. And here also some data, some percentages on the results I got. We were able to get a barcoding effectiveness which was quite high for enziferans with 88%, but also quite low for califerans with 62%. These results are fitting also the results got by a recent, quite an older barcoding project by Aulinsek of 2016. So we expected a low result for califerans. For some reason, I will explain better later. But we also obtained very interesting results concerning the possible existence of cryptic or undescribed species within our dataset, while we also obtained some barcode sharing cases, possibly highlighting the presence of needed synonymization among different taxa. So, as you can see, we got 21 possible cryptic or unidentified species for califerans and 37 for enziferans. Here, I will show you some cases, some example for each case. For example, here we have the case of Tetrix carsi and Tetrix bipunctata. As you can see, all the limitation methods agreed on the limitation of a single cluster for these two species. So according to our molecular data, they should belong to the same taxa, while the morphological data, especially the length of the hindwing, still suggests the existence of two species according to the length of the hindwing. So more detailed taxonomic analysis are needed for clarifying the taxonomic position of these species, of these taxa. While here you can observe the case of possible existence of cryptic lineages within two species. In this case, we have Calyptamus barbarus and Calyptamus sicilia. As you can see, all the limitation methods agree on the presence of two clusters for each species. And I also observed the possible geographical separation between cryptic lineages. As you can see, both species contain two clusters which are also geographically separated. For example, Calyptamus sicilia, which is widespread from the southern of France to the southern of Italy, also including Sicily, contain two different geographically placed clusters, one from Switzerland and the second one from Sicily. And the same thing for Calyptamus barbarus, with a cluster from Greece, Italy, and Hungary, and one from Spain, Portugal, and Switzerland. So further analysis using possibly more markers could clarify the taxonomical position of these possible cryptic lineages. But it's also important to highlight the cases where DNA barcoding is not capable of delimiting the morphologically recognized species. Here we have the case of, for example, of the subfamily Gomphocerinae, where, as you can see, all the limitation methods are able to recognize a single species cluster for all these different morphological taxa, for example, Cortypus, Gomphoceripus, and many more ones. So, in this case, the DNA barcoding is not effective, and a multi-locus approach is needed, like some recently published studies demonstrated, for example, in the genus Cortypus. But why DNA barcoding is not effective for some taxa, for some groups? Why? At first, the most important phenomena is the so-called incomplete lineage sorting, which concern the failure in the consistent sorting of an ancient polymorphism between a new species event occur. This leads to the presence of different evolutionary history between a single gene and the true species. So using a single gene for the inference of the evolutionary history of a certain species can lead to some errors, like you can see in this image. So, and this happen specifically in fast diversification events, like the ones that involved, for example, the Gomphocerinae subfamily. But another important phenomena is the so-called introgression, and in this case, the introgression of mitochondrial DNA, which is the phenomena by which we have the continuous backcrossing of hybrid specimens, animals with one of the parental species. This could lead to the transfer of genetic material from the gene pool of a species to the gene pool of another. So we can get a mismatch between morphological and molecular-based identification of certain specimens. So, in conclusion, the barcoding dataset still provides a good ground plan for the correct identification of several Orthopterans, both califorans and enziferans. But in that cases where the morphological and molecular identifications mismatch, it's important not to use them, at least at the first instance, for the identification of unknown biological material, but it's also important to deepen that cases using further analysis, possibly involving more markers or even a whole genome approach when possible. I also wanted to highlight how the integrative taxonomy can be extremely effective in delimiting the most likely species cluster, like in the case of this study from some Southeastern Asia Orthopterans. And here, some of the next projects I am involved with. We are planning to realize the barcoding dataset of Orthopterans from the Abruzzo, Lazio, and Molise National Park in central Italy, where we will try to assess the presence of recorded and unrecorded species, with the aim to extract DNA and possibly also RNA. And here also my future PhD project involving the evolutionary responses of Orthopterans to the climate and environmental change concerning the genomic data. So we will try to detect possibly adaptive lossy, comparing different populations, and using this data to infer reliable predictions of the future of this population under the threat of environment and climate change, human-driven changes. So these are my team members, which I really thanks for their help, and thank you for the attention.
Oh. Okay.
No, sorry, sorry, the last part of the question. Concerning Podisma, I think we had Podisma specimens, records in the dataset, but they were clusterized into a single species, putative species cluster. Probably the sampling localities for that species were not so numerous, so maybe they were all from the same site and we missed some of the variability of the species. But yeah, there are of course different other phenomena possibly involved in the failure of DNA barcoding delimiting the most likely species cluster. One of them is of course the pseudogenes, but also the new mites, nucleomitochondrial sequences, and as we know, Orthopterans are also mostly affected by Wolbachia, which can cause some problems as well. But it's also important to remark that in the last years, the progress in the design of specific primers for the amplification of, for example, the cytochrome oxidase 1 were able to address these problems in a good way, and in this case we used some specifically designed primers called the COBU and COBL for the amplification of the cytochrome oxidase 1. So yeah, it's right. There are other processes possibly leading to the failure of DNA barcoding in this case. Thank you.
Yeah, just on the case of Punctata and Krause, well, we had started the phylogeny of all the Palearctic tetrachids some years ago, and we had a lot of sequences, mainly mitochondrial ones, so similar problem. But anyway, but I remember that we could not find any difference between Krause and Punctata, while all the other tetrachids were actually quite deep splits.
Yeah.
Yeah, so I suspect that these are just wing morphs, which are also
Yeah.
present in many other Orthoptera. And meanwhile, well, I had some discussions with some Swiss colleagues in the past who were always a bit skeptical about this, but meanwhile they also convinced that the only thing which is really different is just the length of the which is not really very strong character.
Yeah. Yeah, right. Yeah, yeah. No, it's exactly. I consulted some recently published papers that effectively support the synonymization of these taxa. So, I mean, we cannot exclude anything, and possibly more analysis with more markers could be useful, but according to the morphology as well, they, yes, could be just into a specific variability. Yeah. Yeah.
Okay.
So, did you consider to put into your analysis more conservative genes, because the mitochondrial one is highly mutating over the course of evolution. When you add the more conservative genes, you increase the resolution of the data and get more comprehensive results, at least in the group of animals.
Well, yeah. We decided to use the cytochrome oxidase 1 mainly in order also to integrate other public data, for example, the one updated to the BOLD systems. And yeah, there are also other possibly more conservative markers. But yeah, cytochrome oxidase 1, at least for metazoans, is known as one of, according to the results of the beginning of the century, it should be one of the best genes for the limitation of species. But yeah, now that we have the possibility to adopt, to use more markers, several markers, then this could be the best way to correctly, or not correctly, but to delimitate the most likely species. So if I had to redo or maybe deepening some of the taxa I involved, I would probably use, or try to use techniques like the RADSEC or possibly the WGS, where if I have the possibilities. Yeah, thank you.
Specimens have been preserved in some collection.
Yeah, exactly. We have both the dried specimens and
allow to know the specimen from which has been obtained the barcode.
Yeah, yeah. We have all the specimens preserved, and we have some unique codes which can allow us to match the specimens with the extracted DNA. And we also still have the extracted DNA, so we could use it for further analysis in future, and while maybe sequencing more markers or. Yeah.
Okay.
Thank you.
Okay, I think that we finished. Yes.
Yes, thank you, Bruno. It was the last talk for today. I just have some messages because I would like to receive the presentation for tomorrow morning, especially. Sebastian Koenig. Okay. And Zore? Oh, okay, perfect. Thank you.
Tomorrow.
And so, it was a pleasure to see as many of you today, and especially to see that there are also some young orthopterists that are beginning their path on conservation and on study of orthopteroid insects. Well, I hope you enjoyed for today. You are free tonight. I'm not asking anything else from you. And no, not for. We will talk all the night. No, you are finished. Thank you. So you are free to go, and please, tomorrow we will start at 10, but try to be here some minutes before, please.